get started then. I want to tell you all about some work that's been in progress for a few years now to leverage a few new technologies called 3C and High-C to try to resolve the genomes of individual strains in metagenomic mixtures. The inspiration for this work really comes from my colleagues at the University of Wisconsin, who 15 years ago sequenced the first few E. coli genomes. When they finished sequencing those first 3 genomes, the very first thing that they did was to identify all the genes and ask the question, how many of the genes in these 3 genomes are common to these 3 strains of E. coli? But much to their surprise, what they found was that only a small fraction, only about 40% of those genes were common to all 3 of the E. coli, and each one of the strains had a substantial amount of unique gene content, and some of that unique gene content, of course, conferred the phenotype of the strain. So one of these strains is a urinary tract pathogen, the other one causes bloody diarrhea, you don't want to get it, and the other is a commensal non-pathogen. And so if you fast forward to where we are today, the scientific community has sort of discovered that the world, in fact, is teeming with microbes, most of which we don't know how to grow in the lab. And they've invested enormous amounts of effort in microbiome profiling and metagenome sequencing and so on in order to characterize these genomes of the microbes around us. But if you were to apply that to a human and sequence some poo, as lots of people seem to do these days, and try to identify which E. coli is living in that person's gut, you'd be really hard-pressed to make a distinction between these strains, because if you've done 16S amplicon sequencing, the 16S genes are almost identical to each other, and genome-wide, their average nucleotide identity is quite high in the core conserved genes in these strains. And it's not just an academic question, because, for example, the uropathogen here can exist as a gut commensal, and it can predispose women to recurrent urinary tract infection if they become colonized with this bug. So we'd really like to be able to distinguish these strains and resolve their genomes in mixtures. It's not just focused on the human body, of course. There's microbes all over the planet, and strains are important there too. In the ocean surface waters, we've got this dominant organism called Prochlorococcus, and it's a major contributor to global carbon cycling. And the Prochlorococcus exists as a number of closely related strains which differ in gene content. The gene content is thought to confer specific niche adaptations for these different lineages. So metagenomics, of course, has been proposed as the way that we're going to characterize these microbes and understand why they are doing what they're doing in the environment. But the way metagenomics is currently practiced makes this really hard for us. And the problem is that early on in the sample processing, we're destroying some of the most valuable information in these microbial communities. What we do is we take the sample and we smash it, we pulverize it with a bead beater, basically destroying the cells, and that lets us get the DNA out of the cells, but then we shear the DNA again down into tiny little fragments that can go into our next-gen sequencing instrument. And what we've done there is to first destroy the information about how sequences arranged into chromosomes, and then how those chromosomes are packaged up into cells. And so the job of the unwitting bioinformatician is to reconstruct all of that information computationally, but that's turned out to be a very challenging thing for us to do. And so it would be nice if we could change the lab sample processing side just a little bit to give us a bit of extra information or a few more hints about how this genetic information was actually structured in the environment. And so I really like talking about different ways that we can do this. There's a number of approaches I think that could be viable, but the one I want to talk about today is something called High-C. So High-C is a technique for basically using high throughput sequencing to reconstruct 3D chromosome architectures. And I'm sure many of you are familiar with this, either because you've heard me talk about it before or heard somebody else in the cancer genomics community talk about it. But the basic idea is that we use formaldehyde to create these reversible cross-links between DNA and protein. Then basically lyse the cells and allow these DNA-protein complexes, which contain bits of DNA that were in close physical proximity, as cross-link complexes, to then float free in solution. And so they float around in solution and find their own little happy place, and then get cut by a restriction enzyme in the second step of the protocol, which leaves these single-stranded overhangs in the DNA. We then fill those single-stranded overhangs with biotinylated nucleotides, and then do what's called a proximity ligation. And the proximity ligation in these very dilute conditions favors the ligation of free strands that are bound up in the same protein complex. And so what this creates then is this covalently bound complex that we can sequence basically after purifying those ligation junctions. And the data contains essentially a map of which bits of DNA were in close physical proximity at the time of cross-linking. So this is a fairly complex protocol, but people do it because the information is very hard to get in any other way. So we got the idea many years ago that this might be useful for metagenomics, and the premise, of course, is that bits of DNA that are part of the same cell should become cross-linked in the same protein complexes at much higher rates than bits of DNA that are in different cells. So we set out to test this with a little experiment. We created a synthetic microbial community in the lab where we grew up a bunch of strains that we had reference genomes for. And then we mixed them and applied the high-C sequencing protocol, sequenced it on MySeq, did metagenomic assembly, and then we mapped the reads, those high-C reads, back to the contigs and asked basically which contigs were being linked to each other with these high-C reads. Now, if you represent this as a graph, a contig graph, or a scaffold graph, it looks a little bit like this. And if we just do a standard layout using a graph visualization program, Getty, you can already start to see some structure emerging here. And of course, then if you color those nodes by the organism that they came from, you can see that the clusters, the structure is quite clear. It's grouping bits of DNA and contigs that came from the same organism. So can we basically use a clustering algorithm to automatically pull these clusters out? And the answer turns out to be yes. After a bit of fiddling around with parameters, we were able to get Markov clustering to produce 4 clusters, each of which contains over 97% of the gene content from each of the 4 species that went in. Now you'll notice here there's this big red blob with lots of contigs. Those are the 2 strains of E. coli that went in, and they were pulled in together into the same clusters, so they weren't actually separated into separate clusters, and we're going to come back to that in a little bit. So this was really exciting for us. We wondered why it was working so well, and the bottom line is that these data essentially look like giant mate pairs. You get pairs of reads that can span the entire chromosome of these organisms in 3C and high-C data. You also, it turns out, get read pairs that couple plasmids that were part of the same cell back to their host chromosome. And so this is information that would be basically impossible to get with long-read sequencing, for example. And those plasmid host chromosome links are appearing at several orders of magnitude higher rates than background linkages or noise in the data. And so that's what's shown in the level plot on the right side of the slides. So this is exciting, and being obsessed with strains and population genetics as I am, I started wondering if we could resolve strain differences. And to do this, I looked at single nucleotide variant graph. So basically, you build a graph which contains as nodes all of the single nucleotide differences among the 2 E. coli strains. And then I just asked a really simple question about the connectivity of that graph. what is the size of the largest connected component in that graph when you link up the nodes using these high C and 3C read pairs. And it turns out that with high C, you get nearly the complete set of nodes in a single connected component, which is fantastic because that might give you information to resolve the genotypes. With mate pair data, you get less in the largest connected component. And that makes sense, of course, because high C is making global connections in the graph, whereas mate pair data was only able to make connections as far as the insert size. We'll take it. And so if you then look at the sort of path length to traverse to various distances around the chromosomes, you can see that as expected, that path length grows in mate pair data, but is essentially flat with high C data. So these results were independently reproduced by a number of other labs. This group, led by Jay Schender and Maitreya, had a really nice paper that appeared around the same time as ours. Excuse me. Applying this to yeast. Sorry. And they got even better results than we did, as a matter of fact. Now, these protocols, as I mentioned earlier, are quite complicated, and there was a paper that appeared a few months after ours that introduced some nice simplifications to these protocols. This is called metagenomic 3C, or meta 3C. And the main differences are that instead of using restriction enzymes that have a 6-base recognition site, they use 4-base recognition sites. And they skip the biotinylation enrichment and just sequence the DNA right after proximity ligation. And surprisingly, this actually works. You can get enough proximity ligation read pairs in the data without that enrichment step to be able to do some pretty useful things. And so when they initially published this protocol, they didn't actually publish the data to go along with it, which was a bit worrisome. I'm surprised they were allowed to do that with eLife because it's against journal policy, but we took it upon ourselves to independently reproduce this protocol. And so Michael Liu in the lab here did this on a synthetic community, and we released the data immediately, and it was published sometime after. And it does actually work. It was able to work in our hands. Confirmation of this came in the form of us reconstructing 3D structures of a few of the bugs in the mixture. So what you're looking at here are 3D structures of E. coli and Pseudomonas aeruginosa. A genome structure is kind of circular donut-like my favorite food. And Pseudomonas aeruginosa has a genome where the 2 chromosome arms between the origin and terminus are intertwined with each other, so it's kind of twisted and snake-like, if you will. And the data is in this NCDI session if you want it. So, given that we can generate this data, how do we go about using it to resolve strains? I think we're going to have to invest a fair bit in new computational methods to do this. But just speculating about how it might be done, you could imagine starting with a content graph or a string graph, which might look something like this. And layering the proximity ligation read data on top of it basically is weighted links among the nodes. And so using those weighted edges, you might use either simple clustering algorithms, as have already been applied, to try to resolve strain genomes. Or you might do something a little bit more clever, like compressive sensing, or apply stochastic block models, or even some of the Bayesian community detection algorithms that have really started to take off in social network analysis. Matt DeMare, who's a student here, has taken it upon himself to try to understand how these different clustering algorithms behave on metagenomic 3C data. And because we don't have a whole lot of real data available, we've taken a poor man's approach to this, which is to essentially simulate large numbers of metagenomic 3C data sets. And the way this works is basically to start with a defined phylogeny, which has just 4 tips. They're all closely related. They're meant to be strings of the same species. And we use a forward time simulation of genome evolution to arrive at a collection of genomes that are between 85 and 99.5% nucleotide identity. We then mix these at different abundances into a community and simulate genome sequencing of them. We simulate metagenomic 3C read pairs, or IC read pairs at different depths. And then we conduct an assembly. We're not exploring different assembly algorithms, just a single algorithm there, single mapping algorithm to map those IC reads back to the assembly. And then we looked at a few different clustering algorithms, Markov clustering, something called O-cluster, SRMCL, which is soft regularized Markov clustering, and then two flavors of Levan clustering, the basic Levan algorithm, and a very trivial extension of that to generate soft clusters which overlap each other. And the rationale behind doing that, of course, is that when you've got multiple strains which share some gene content, you would expect in some cases that the shared gene content might co-assemble into single contigs. And so those single contigs, if you're doing a clustering, might need to be a part of multiple clusters in order to accurately reflect gene content in those genomes. And so the trickiest part of this process is the last bit, the validation, of course. And so I'm going to talk a little bit about that. But before I do, I want to tell you what the assembly graphs look like. So when we simulate these assembly graphs, we found basically that there are some really big changes in the properties of the assemblies as a function of the average nucleotide identity of the simulated strains. And I guess some of this is to be expected, although the details of where these changes take place were a little bit surprising to us. But basically, at the one end, when all of the strains have relatively low sequence identity, 85% average nucleotide identity, we found that they basically assemble entirely separately into high contiguity genomes. So you get each of the strain genomes in separate contigs. But as that nucleotide identity goes up, the size of the contigs deteriorates down to a critical point around 98% identity, when the contiguity actually starts to go back up again. And that, of course, is because the strains are now so similar, about 98% identity, that the assemblers are just squishing and co-assembling all of the contigs together. Surely there's different assemblers do different things. The only one that we looked at was IWAD. The graph complexity, which we've measured using an entropy measure, H sub L there, changes in a similar manner where it kind of goes up until it reaches a peak, and then at very high identities, assembly graph complexity goes back down. Okay, so how do we then measure the accuracy of clusterings on these contigs? Well, this is tricky for two reasons. The first being that an individual contig may be a product of multiple different genomes. And so when we make these assemblies, we lose track of which reads went into the contigs because the de Bruijn graph assemblers don't give you that information. So we instead used a pairwise sequence alignment approach using LAST to map the contigs back to the reference genomes and build up a gold standard using that. And the details are in a preprint that Matt has up on PeerJ now, if you care to get into this. So we build up this gold standard of mapping of contigs back to the references. And once we have that, then we need a metric to measure the accuracy of clusterings. There's a bunch of different ways that you could do this. We settled on something called extended B cubed, and then further extended B cubed to weight the answers by contig length, basically. And so we chose this because there's a number of nice properties that extended B cubed has. I'm showing those four properties on this slide. Basically, it favors more homogeneous clusters. It favors more complete clustering, so you want all of the elements in a single cluster if possible. It tends to favor smaller sets of clusters with fewer clusters in them. And it supports this notion of a RAG bag, which is this idea that if you've got a bunch of random elements that you can't cluster very well, rather than spreading them among a bunch of other clusters, you'd like to lump them all into a single ragtag, effectively. And so applying that measure, which actually has a notion of precision and recall, we get results like this, where what you're looking at is on the top series, the precision, and the bottom series, the recall of each of these 5 clustering methods as a function of the average nucleotide identity of the genomes that we simulated. And there's some pretty straightforward conclusions that can be made from this. Some of the clustering algorithms are much more precise than others across the range of parameters that we looked at. But in the recall, it's kind of interesting. Pretty much all of the algorithms suffer in the recall department as this average nucleotide identity goes up. And the only real exception to that is this very trivial extension to the Levain clustering algorithm, where you can see that as we add more 3C read pairs to the data, the recall gets a little bit better, but still not great at high average nucleotide identity. Right. So to try to get a little bit more insight into this, we calculated the F score, which is, of course, just the harmonic mean of precision and recall. And we did some multidimensional scaling, or PCA, on this. And we took basically all of the scores for all of the different algorithms across all of the different simulation configurations. And did this dimensionality reduction and asked essentially what factors were influencing or correlated with the accuracy. And so what we found there is that the average nucleotide identity, of course, is negatively correlated with accuracy of most of the clustering algorithms. The big exception, as I mentioned earlier, is the soft Levain algorithm. And you can see how that one the vector for that heads up on the second principal component, and there's some correlation there with the amount of 3C data that's been generated. And so that suggests that perhaps we didn't even simulate enough 3C data for that algorithm, that the results could have improved further in some of the high strain identity scenarios. So everything I've talked about so far has dealt with simulated data, which is kind of fine, except that it's not very practical. We'd like to be able to do some real biology with this approach. Now, data on real samples has been quite slow to emerge, unfortunately. But there was a publication that came up very late last year, just in time for Christmas, from a group at Pasteur, where they applied this protocol to mouse feces. And this is essentially the first application to a natural microbial community. And so it's quite interesting from that perspective. What they did was to generate about 30 gigabase pairs of Illumina reads, very short ones, 65 base reads. That produced a 580 megabase pair genome assembly with an N50 of around 4 kilobases. And they applied Levain consensus clustering onto this, which resulted in 178 communities that were between 1 and 6 megabase pairs in size, so your typical bacterial genome size. And then they took those individual communities, those clusters, and they scaffolded them using the software called Graal. And if you then take one of those, or these 3 of them in this slide, and look at the heat map for the 3C read pairs, we can actually see now the 3D chromosome structure coming out in these. And that's apparent as this sort of anti-diagonal that crosses the main diagonal in these heat maps. So that's indicative of a circular chromosome where the 2 chromosome arms, are interacting with each other. So that's really exciting. So will we be able to look at strains and shared gene pools in this data? They took a very cursory attempt at this in this work. they ran 5 iterations of the band clustering and made use of stochastic tie-breaking behavior to get different clusters out of each run. And then they asked how frequently the same contigs were basically clustered together across the different runs. And so that's what this plot is showing. And what you can see here is that there's, for a given sort of larger area that's got heat, there's multiple sort of cores that are associating with what appears to be ancestry gene content. And so potentially we've got either multi-chromosomal genomes or a few different strains present in the data. But I think it needs a much closer look to resolve what's really going on here. So where to next? I'd like to say that we were much further on with this work than we are right now, but unfortunately we're not. I think we're being held back by wet lab protocols. We need them to be easier and better so that we can generate more real data. On the computational front, I think we really need to work to translate some of these accuracy metrics into a more accessible framework. And so it'd be great to talk about whether that'd be possible in the CAMI framework. One of the next things that Matt has been doing is to migrate some of this parameter sweep code into something called Nextflow. It's a domain-specific language for workflow management. So that's something that you could talk to him about if you're interested. But with that, I'll end. Got a lot of people to thank, especially the people here at UTS who've done most of this work, because I did very little of it. I just get to talk about it. Of course, Alice and Alex for heading up this great initiative that we're part of. So thanks, everyone. Oops.
Thank you, Anna. Aaron, with your point of how to integrate strain level identification into CAMI, do you mean using these kind of data sets, or something in addition?
Well, I think data sets like this are maybe nice for engineering tools, but when it comes time to evaluate and compare tools, it seems like it would be nicer to have real data.
Yes. So how would you design data sets for a challenge? Or how would you generate them?
Yeah, that's a great question. I mean, one thing that you could do would be more of the same, where you make synthetic microbial communities. But perhaps you could do that with organisms that are not yet in public databases. Another possibility might be to generate data sets like this. You'd have to work very closely with microbiologists, but perhaps they could cultivate some of the organisms from the community and get a few new things.
Okay. Do you think? I mean, how many people would be like for a challenge? How many people would be able to? Or how many tools would be able to work on those kind of data sets? Is it? Do you need to
Almost none. It's really a niche right now. I don't think it's had very wide use in the scientific community yet.
Do you think it would be possible, or a difficult extension for standard tools to work on that kind of data?
Possibly. Scaffolders, for example, make their data and create collections, but they've got very strong assumptions about the insert sizes of reads, and so it's not a straightforward extension. I think with strain evaluation in general, the covariance-based techniques have a lot of potential to resolve strains as well, and so if there was going to be an effort into strain resolution in the CAMI context, it might make more sense to use that as the starting point rather than 3C data, which seems like it's still pretty rare in the wild?
Yeah, I mean, we've had some strain, and we have results in part for strain level resolution shown yesterday. What was your thought on those? I think you watched some of
Yeah,
them.
I listened to Alex's talk. Is that what you're referring to?
Mm-hmm. Yeah, yes.
Well, there was no resolution for strains, basically.
That's what I got from it too.
I mean, the binners, the covariance-based binners for the low complexity data sets seem to be the most promising thing. So they had the genome resolution, and that was shown across, well, for all genomes, including those that are just simple strains or not. I mean, it wasn't resolved further so far, admittedly. So I guess maybe we still have to investigate a bit more how the performance of the binners depends relative to whether it was a multi-strain species or a single strain species in a data set.
Sorry I missed the your binning talk yesterday, unfortunately. Sorry, I didn't see, but was the is it possible for a single assembly config to go to multiple bins?
Sorry, for a simple, what was that?
Is it possible for a single assembly config to be assigned to multiple bins in the CAMI framework?
One config in different bins? No.
No, I think it's always one bin.
One config, one bin? Yeah.
You're thinking of some kind of probabilistic assignment, or why?
Well, just that if you've got both the related strains, some of those configs will be co-assemblies of content from both strains.
Yeah, that's right. Yes, we talked a bit about that. That this is probably this is a direction for a problem for assemblers to tackle, right? In the output.
Yeah.
Yeah, and maybe also for us in our evaluation measures. That's a good point. You had several suggestions on how to, integrating measures into CAMI. I think that would be very welcome. Peter Bellman, I think at the moment there's already a template for evaluation Docker containers. Have you seen that?
Yep.
So is that like what you were thinking of with that point?
Yeah, I think that'd be a great place to start.
Okay. Yeah. Assembly really, the question is which measures are most useful. So we had this discussion yesterday as well. Are you aware of those? What's coming out of what is this tool called that we're using? The MetaQuest.
MetaQuest.
Do you know those MetaQuest measures and for assembly evaluation?
Yeah, well, that's what you were discussing yesterday, Alex. Yeah.
Yeah. The question is what's most useful of those measures, or if, as you say, maybe we need different ones in addition.
Yeah, the measures that I was discussing today really only look at gene content or contate content assignment to strains. So it's pretty limited in that regard. I know MetaQuest has many other metrics which are going to be useful for different things. I think this is a case where beauty's in the eye of the beholder, and it's really going to depend on what you want to do next with the data.
Yeah. We need to, I think, for the future CAMI contests, we need to work out more clearly, really, for which specific applications the results are supposed to be good, and which measures will be most important to assess them.
Yeah, I mean, the dimensionality reduction might help with that a little bit. It was something that we applied in the assemblathon simply to understand which metrics were highly correlated with each other.
Yeah, that's very nice. I agree. Yeah, we thought about that, but we didn't manage to finish all on time. Okay, well, thank you very much. Aaron, if you like, we have just these working group discussions later on the future of CAMI. So if you want to contribute, we can kind of get you involved. I'm sure in some ways, maybe via Skype. Or if you ever want to fly around half the world again to join us in person, let us know. Would be happy to have you.
I hope I get the chance.
Yeah, it'd be cool. So thank you again for your talk.
All right, thank you all very much. Thanks for having me.